So in our previous lecture, we learned about humoral immunity and the long and complicated process that ends with the synthesis of large quantities of antibodies that target a particular antigen. Antibodies are incredibly effective as long as the antigen is in the extracellular fluid. But what happens when you have a pathogen that invades our own cells? These are intracellular pathogens. All viruses have the capacity to invade our cells. In fact, it's required by their life cycle. And it turns out that some bacteria can do it as well, including, but not limited to, bacteria that cause gonorrhea, tuberculosis, and Lyme disease. So, when we have a virus or any other intracellular pathogen, humoral immunity is part of the solution, but it's not the whole solution. We need another way to deal with our own infected cells and the pathogens that are in the infected cells. This is known as cell-mediated immunity. The two major players involved in cell-mediated immunity that we have not yet been introduced to are going to be cytotoxic T cells and MHC I. So let me start with MHC I. MHC I is capable of being produced by all nucleated cells in the body. This is quite different from MHC II. Remember that MHC II is produced only by macrophages and B cells and also dendritic cells, but I don't cover dendritic cells, so for my purposes, it's just macrophages and B cells. Here, basically any cell in the body that has a nucleus can make MHC I. And on that note, can you think of a cell type that doesn't make a nucleus? You should. Red blood cells. So red blood cells are an exception to this because they do not have a nucleus, but most cells in the body do, and therefore are capable of making MHC I. MHC I, like its cousin MHC II, is a display case for antigens. However, unlike MHC II, MHC I is not a trophy case. In other words, when antigen is inserted into an MHC I case and displayed on the surface of the cell, it's not a trophy. It sends an entirely different message to the immune system. It asks the immune system to come and check out what's displayed on the surface. And if what is displayed on the surface is antigenic, it will lead to the destruction by apoptosis of the cell that actually displayed it. So in this way, we think of this sometimes as kind of like a suicide signal or a potential suicide signal. So the immune cell that responds to an antigen in MHC I is called a cytotoxic T cell. Okay, let's take a look at how this works. We are going to assume that we are infected with a COVID virus. Now, the way viruses work is they use some protrusion on their surface, such as a spike, to bind to some sort of receptor expressed by a cell. And cells in the back of your throat and in the back of your nasal cavity express large numbers of receptors that COVID likes to bind to. So when it binds, this actually triggers the infection. So binding of the virus to a receptor on one of our own cells is actually what permits the virus getting into our cells in the first place. Once the virus is inside of our cell, its goal is going to be to make a bunch of new viruses. Now, I'm being a little bit simple here in my explanation. Viruses don't replicate like cells, so it's not as if you take one virus and you split it into two. Rather, you have to make the individual parts and then put them all together. So I'm not being entirely truthful in how this works, because it looks like the viruses come pre-assembled, and they don't. You have to make the parts first. But what that means is that we're also making a bunch of these spikes. When the cell makes large quantities of anything, it will insert those into MHC I and then display MHC I with that potential antigen on the surface of its cell. So here, and let's take a look at what happens next. The antigen MHC I complex is actually stored up in vesicles, and then we have exocytosis, where these vesicles are brought up to the surface of the plasma membrane, like so, and then allowed to fuse, like so. And this basically allows that MHC I with the antigen to be displayed into the extracellular fluid. So let's assume that we've gone ahead and put these on the surface, like so. So question, is this now an antigen-presenting cell? You might be tempted to say yes, but the answer is no. We only use the descriptor antigen-presenting cell, or APC, when we are dealing with MHC II, which is a display case analogous to a trophy case. Here, this infected cell is not bragging. It is alerting the immune system that something is potentially terribly wrong inside, and the immune system needs to come and take a look at it. So the responsive cells to antigen in MHC I are cytotoxic T cells. And cytotoxic T cells are shaped very similarly to helper T cells. They're small, they're lymphocytes, and they have a T cell receptor. The T cell receptors have specific cutouts. And just like helper T cells, cytotoxic T cells are going to have a T cell receptors that binds to antigen in an MHC case. In other words, the T cell receptor cannot bind to free antigen. It can only bind to an antigen in an MHC case. And at this point, it's useful to differentiate between the helper T cells, T cell receptor, and the T cell receptors found in the cytotoxic T cells. So let's go ahead and differentiate them. So as a general rule, cytotoxic T cells have T cell receptors that bind to antigen in MHC I only. They don't respond to antigen in MHC II. Conversely, helper T cells have T cell receptors that bind to antigen in MHC II. In both cases, we need that perfect puzzle piece fit. We need the cutout shape of the T cell receptor to match the antigen in the MHC case. Just like helper T cells, there are many cytotoxic T cell clones, and each clone differs in the cutout shape of its T cell receptor, as you can see here. So when an infected cell is displaying, and notice I'm not using the word presenting because it's not an antigen presenting cell. So when the infected cell is displaying the antigen on an MHC case, then that calls the attention of cytotoxic T cells. So we might try a cytotoxic T cell comes over, no fit, and goes on its merry way. Another one comes over, and we have a perfect fit. So once we have a perfect fit, if the cytotoxic T cell is active, the cytotoxic T cell will induce apoptosis in the infected cell. Let's take a look at that sequence of events. Okay, The first thing the cytotoxic T cell does is secrete a molecule known as perferin. And perferin is actually like a little pore that is going to insert itself into the plasma membrane of the infected cell. So we get these little pores. They're not enough to cause lysis of the cell. And in fact, we do not want to lyse the infected cell. If we lysed the infected cell, the viruses would be allowed to go everywhere into the extracellular fluid. And if there were 1 million or so viruses in here, that would be too much for our immune system to rapidly clean up. And so we want to make sure that we contain the viruses rather than letting them leak out through an exploded or a necrotic cell. So we do not actually want our infected cell to lyse. Instead, we want to induce apoptosis. So the next thing that cytotoxic T cell is going to do is send a variety of signals through that small opening into the nucleus of the cell that is going to activate the process of apoptosis. So apoptosis is a pretty involved process. It's not a one-step process. It's got a lot of moving parts to it. And we have genes in our nucleus that are turned on or activated to basically trigger that sequence of events. And the cytotoxic T cell's endgame is to ensure that the infected cell turns on the apoptosis genes. So there are names associated with these signals, including, I think, granulozyme, but they don't make a lot of sense in terms of what they do. So I'm just going to collectively call them the apoptosis signal. Okay, and a cute way that you can remember the word perforin, if you're familiar with perforated paper has small holes in it. So perferin makes small holes. I think that's kind of a cute name for that particular pore, so I like using it because I think it's easy to remember. All right, so now that we've looked at the apoptosis signal, that is going to induce apoptosis of this particular infected cell. So let's quickly go ahead and draw this. So remember, apoptosis is going to break the cell up into small blebs. So these blebs are actually going to contract some. Okay, and so now that we've successfully achieved apoptosis, what we need to do is clean up all the viruses, which are still contained but may not stay contained indefinitely. So the cytotoxic T cell's final farewell or swan song here is to secrete cytokines that call over phagocytes, including macrophages. And so now what's going to happen is these big macrophages are going to come on the scene, and they're going to try to eat up all of those blebs and finish the process. So that is how we deal with infected cells. We trigger apoptosis, and then we clean up the mess, and hopefully the viruses are still contained. We'll be adding a little bit of detail to this particular story in the next lecture, but this is enough to get us started, and we have a basic idea of the main steps that occur in cell-mediated immunity.